Hello, my name is Kristina Ryabovol, and I will be talking about fluorescent indicators to track protein interaction and maturation. To conduct my research, I worked with the Picower Institute at MIT. The interaction between proteins is the foundation of biological research. The ability to see chronology of protein maturation and interaction provides important insight into the context-dependent processes. By creating the ability to see long-term the maturation status of proteins through the photoconvertible EOS protein and the active interaction between proteins using NEMO, a full picture of biological functioning could be extracted. Through creating a reversible interaction between gyrin and BRP for the NEMO fluorescent, the same larva can be used repeatedly. The combination of these findings allows researchers to understand why and how their interactions occur, and most importantly, when. Recent advances in fluorescent-based microscopy allow scientists to detect fluorescence of single molecules. This fluorescence enables them to detect protein positioning and interactions, thus addressing key questions in biological functions. Current fluorescent detectors are able to accurately detect spatial interactions of fluorescent molecules, however, temporal protein stage and interaction is still challenging. The major limitation for application of currently developed sensors is that they either are irreversible or have an extremely weak signal that can only detect interaction between large protein clusters. We tested the photoconvertible protein EOS as a possible receptor for the protein age and used reversible splittable fluorescent protein NEMO as a possible detector of protein interaction. The protein NEMO is split into M-neon-1 and M-neon-2 to detect protein position and age. Genetically encoded fluorescent markers fuse with the protein of interest. In this research, we first attempted to characterize the chronology of protein accumulation at the neuromuscular junction. Genetically encoded fluorescent marker EOS was fused with BRP and photoconverted in live Drosophila larvae at the early stage of development. We were successfully able to detect distribution of old and new proteins at the same animal in later stages of larva development. Second, we tested the functionality of the fluorescent protein NEMO, which was fused with BRP and gyrin. We characterized the temporal aspects of fluorescent marker NEMO and are currently in the process of testing it in protein interaction. We used the Drosophila larval neuromuscular junction as a model system. Motor neurons exit the Drosophila ventral nerve cord (VNC) and projects and innervates the muscle. In the area of the contact between motor neuron and muscle, specialized structures formed called active zones. This is the area where the synaptic transmission occurs. A protein BRP accumulates in those areas and positively correlates with synaptic transmission. New fluorescent proteins were tested via creation of genetically encoded fluorescent markers attached to the BRP. In the Drosophila neuromuscular junction, there is a key protein BRP localized at the area of synaptic transmission, active zone. The second protein, gyrin, localizes at the membrane of synaptic vesicles. Maturity of the protein BRP and the interaction with the vesicle are key factors defining the functioning of the neuronal release site. The first sensor we worked with was EOS. EOS is a photoconvertible protein. It irreversibly switches, which means that fluorescence emission spectrum upon illumination with ultraviolet light. So its irreversibility is defined by photo-induced modification involving a break in the peptide backbone near the chromophore. This break causes the protein to change its strong green fluorescence to red upon UV irradiation at 390 nanometers. We used it to track protein maturation and turnover rate, as well as test the brightness and stability of this fluorophore. To test protein maturation, early instar larvae expressing BRP tagged with EOS were illuminated by the fluorescent light of the 405 wavelengths. This triggers the transition of fluorescent markers EOS into the red state. Therefore, it is expected that all active zones carrying BRP protein will be labeled with the red color at this time point. A schematic representation of this can be seen here. Dissection of the larva before photoconversion demonstrated that all proteins existed only in a green state, confirming that no spontaneous transition into a red state occurs under normal light conditions. Photoconversion was attempted for 15 seconds, and preparations were dissected immediately after photoconversion. No residual green fluorescence was seen after 15 seconds of illumination with 450 nanometer wavelength, proving that it is sufficient to photoconvert the entire pool of the protein. Some larvae were dissected only 2 days after photoconversion, and combination of old and new arriving protein at the active zone was observed. The majority of active zone was photoconverted, and the signal of old photoconversion active zones was also observed. These results confirm that photoconvertible proteins can be used as a marker for protein age in a live animal. The second sensor used was NEMO. NEMO is a reversible splittable fluorescent protein, which means the fluorescence of the sensor NEMO is based on splitting the fluorophore M neon into 2 parts. The original research demonstrated that the reconstitution of the full protein provides bright fluorescence that is reversible upon further splitting of this protein. Traditionally, split-based fluorescent systems were based on splitting another protein, a GFP, but its recombination was irreversible. We used it as a calcium sensor and tested its temporal fluorescence pattern and reversibility of the signal. The ability of NEMO sensors to report transient fluorescent events was first tested by expressing the entire sensor in a postsynaptic cell, where it detects calcium influx through glutamate receptors. Indeed, transient fluorescent flashes were observed postsynaptically, confirming that NEMO can serve as a detector of transient protein interactions. NEMO was placed on the BRP and connected by comodulin and NEMO2. When there is a calcium influx, the 2 parts of NEMO would be brought together, and thus a fluorescence would occur. Here is a neuromuscular junction before stimulation where there is little photoconversion. During stimulation, 500 milliseconds after stimulation, and 1 second after stimulation. We can see that it is reversible because after stimulation, the fluorescence died down, and during stimulation, there is bright fluorescence. In the future, temporal characteristics of NEMO sensor should allow to track interaction between different proteins. Since the sensor has a minimal background fluorescence when in splitted stage, it should provide a strong signal only when proteins interact, as it did here. In the case that the proteins depart from each other, we expect that within 1 second the signal will diminish. Currently, we are testing a model where one part of NEMO is attached to BRP, while another part is attached to gyrin through the vesicle. This study demonstrates the use of fluorescent microscopy to detect protein interactions and their maturation in living organisms. The EOS photoconvertible protein was able to show the maturation of proteins, ensuring the crucial temporal aspect of protein dynamic can be detected. The visual temporal changes in proteins of the same Drosophila larvae present this as a valuable method for tracking protein maturity. The split fluorescence system linked to BRP and gyrin offers a novel method of tracking real-time interactions. Collaboratively, these methods offer a framework for comprehending protein interactions and time of development. This work is fundamental to analyzing temporal synaptic protein functions and protein interaction. Thank you.